The overall goal of this methodology is to analyze navigational behavior of Drosophila larvae in response to simultaneously optogenetic stimulation of its olfactory neurons.
This method enables the comprehensive dissection of olfactory circuit function and complements studies on how olfactory circuit function translates into behavior response.
The main advantage of this technique is its specificity and temporal control by using light instead of traditional odor gradients to activate individual olfactory neurons.
To begin the experiment, build a light-deprived behavior arena. Construct a box that is 89 by 61 by 66 centimeters made of black-colored plexiglass acrylic sheets, 3 millimeters thick. Place the box on a tabletop in the behavior room. Next, mount a monochrome USB 3.0 CCD camera fitted with an IR long pass 830 nanometer filter and an 8mm f1.4C mount lens on the center of the ceiling of the black box. Place 2 infrared LED strips on the tabletop to illuminate the larvae in the dark arena. Then, obtain a 22 cm by 22 cm square aluminum plate to build the LED platform. In the center of the plate, use a metal cutter to cut a hole that is large enough to fit around the CCD camera. Cover the metal plate with red LED strip lights. Solder the LED light strip wires in series and feed the strip wires into an optocoupler relay controlled by a Raspberry Pi 2B microprocessor.